Welcome to the part 3 of revision series of human physiology, neural control and coordination chapter. In this video we will cover human eye and human ear, but before that let's cover some basics of sensory reception and processing. First, as you know, smell, which is also called olfaction. The sensory organ of smell is nose. Nose has mucus coated olfactory epithelium, which you must have already studied about in the animal tissues chapter. Now this epithelium has three types of cells. One is basal, then supporting, then olfactory receptor cells. We will learn about this later. Next, now these olfactory receptor cells, they extend back to CNS and connect to something called olfactory bulb, which are extensions of limbic system, which you must have already studied in your last video. That limbic system also helps in olfaction. Now next part, moving to the role of brain in olfaction and gustation. So now olfaction and gustation are both involved in sensing dissolved chemicals. Gustation through tongue and olfaction through nose. Now the flavor is perceived by mixing olfactory and gustatory stimuli in the CNS. There is something in brain called the primary gustatory cortex, which is located in the parietal lobe, and it is responsible for perception of taste. Next, moving to the human eye. This diagram of human eye you must already be familiar with. If not, you can pause the video and once again revise all the structures. The things that I have marked with a cross that are not in NCERT and are not important. For example, Sheem's canal is not important, but all the other things are important. The things that are marked in green are very important. Now moving to the functions. First, in the eye there are two chambers. There is this aqueous chamber and then there is the vitreous chamber. So this aqueous chamber is present between the cornea and the lens, and it has aqueous humor, which is a thin liquid which is transparent. And then in the vitreous chamber, which is present between the lens and the retina, they have vitreous humor, which is a transparent gel. Now moving to the functions of different structures in eye. There are three layers in eye. First there is the external layer, there is then internal layer, and then there is the middle layer. So as you know that I have marked the external layer in blue, middle layer in yellow, and the internal layer in green. So starting with the external layer, so there are two parts in external layer. One is the sclera, which covers this portion of the eye, and the cornea, which is present in front of lens. So this both sclera and cornea, they are both made up of dense connective tissue. You must remember which type of tissue they are made up of. They are made up of dense connective tissue. Now this middle layer, it has three main parts. First is the iris. As you can see, this iris, it is anterior, opaque, pigmented and visibly colored portion of the eye. Now this iris, this regulates the diameter of the pupil through muscle fibers. Next, there are ciliary bodies which are present over here. Now ciliary body hold the lens in place through ligaments. They don't use muscles, they use ligaments. Next, there is choroid. Now this choroid, what it does is, it is mentioned in NCERT that it is thin in the posterior 2/3 part. So in this part it is thin, and then it gets thicker in this part. Now it may contain blood vessels and etc., and it functions in providing blood to the structures of eye, and it appears bluish in color. Next, coming to the most important parts of the eye which are present in the inner layer, there is of course the retina, which is the screen. Now retina contains rods and cones cells. It also contains bipolar cells and ganglion cells, which we will look into later. Next comes this macula. Now macula lutea is yellow pigmented spot laying at the posterior pole of the eye. Now this spot, what it does is not important, but it has a central pit called fovea. Now fovea's function is important. So fovea only contains cones which are densely packed. They are present in the thin central portion of retina, and this is the point where visual resolution is actually greatest. So in the macula, which is the pit, there is a point called fovea, where the visual resolution is greatest. And also somewhere over here there is a blind spot in which there are no cones and rods, and over here you cannot see anything. If the light falls upon the blind spot, you cannot actually see anything because there are no rods or cones. Remember, in fovea there are only cones which are densely packed. After that there is this optic nerve which takes the signal to the CNS. These are the important parts of eye. As you can see now, this retina, it has three types of major cells. This retina, first there are bipolar cells, ganglion cells, and over here are the rods and cones, and this is the retinal pigment epithelium, RPE, which is present over here, and from over here goes the optic nerve. Activation of these bipolar cells and rods and cones create an inverted image in the retina, which is picked up by the optic nerve and sent to CNS. Blind spot we have already seen. Now here it is the mechanism of the vision. But before mechanism of the vision, let's first just go over this rods and cones. So as you know, rods, the vision perceived by rods is called the twilight or scotopic vision. They have a photopigment called rhodopsin, which is a purplish reddish protein. It is also called visual protein, purple. Now the cones, they are responsible for daylight vision. Rods are responsible for nocturnal vision, and cones are responsible for daylight vision, which is also called photopic vision and also called colored vision. There are three types of cones, which is green, red, and blue. Now different photopigments are present in these cones. Sensation of different colors is given by various combination of these three primary colors. Now when these are equally stimulated, white color is perceived. The photopigments contain an aldehyde of vitamin A or retinol and a protein called opsin. How that functions we'll see in the next part, which is the mechanism of the vision. So first, as you know, visible light enters the cornea, which passes through lens, and then lens focuses the light rays and passes it to the several different photosensitive cells. Now these photosensitive cells, as you have already seen, contains rhodopsin, which is an aldehyde of vitamin A, and this rhodopsin, which is a photopigment which contains an aldehyde of vitamin A called retinol and a protein called opsin. Now what happens is that this rhodopsin, it breaks into two different structures. It breaks into opsin and retinol when it is activated. So it breaks into this, as you can see, this aldehyde of vitamin A is retinol and this protein opsin. As light hits this rhodopsin, it breaks into retinol and opsin. Now this breaking of rhodopsin into retinol and opsin in the photosensitive cells causes change in membrane permeability, and this generates potential difference in photoreceptor cells. And through this photoreceptor cells, it is transported to the bipolar cells, and from the bipolar cells it is transported to ganglion cells. From ganglion cells it is transported to the optic nerve, and from optic nerve this impulse moves towards the visual cortex. That was the functioning of human eye, the endomechanism of vision. Now let's move to the human ear part. Human ear is anatomically divided into three divisions. There is first outer ear, which contains both pinna and ear canal. Then there is the middle ear, which contains malleus, incus and stapes, which are the three bony structures present in ear. And then there is an internal ear, which is also called inner ear or labyrinth. Now outer ear, pinna and ear canal, it has several components. It has fine hair and wax glands, and then this pinna, what it does, it gathers and collects the vibration in the air and passes it through the external auditory meatus. External auditory meatus takes all the vibration in the air and passes it to the tympanum or the eardrum. Now, tympanic membrane, it is composed of three bony structures or ear ossicles which are arranged in chain. They are malleus, incus, and stapes. So malleus is attached to the tympanum, and stapes is attached to the oval window, which we will see later, oval window. And then incus is between them, attached both to malleus and stapes. Now the function of the ear ossicles is that they increase the efficiency of transmission of sounds. Tympanic membrane is a connective tissue covered with skin on the outside and mucous membrane in the inside. This also helps in increasing the efficiency and transmission of the sound. One another important part of human ear, as you may remember, that human ear is not only a sensory organ for auditory stimulus, it is also a balancing organ, and that balancing is done by vestibular apparatus, which is present in the labyrinth. Now this labyrinth is actually the fluid filled inner ear. As you can see, the blue part is involved in the balance, and we will go into that later. And this part, basically cochlea, is involved in hearing. So the structure which is shown in blue basically plays part in balance. It is fluid filled and it has two parts, bony and membranous labyrinth. The bony labyrinth, which is filled with perilymph, encloses the membranous labyrinth. So there are parts of membranous labyrinth which are vestibular apparatus, which is a complex system. It contains semicircular canals. There are these three semicircular canals which are organized in right angles to each other, and they have receptors called crista ampullaris. And then there is the otolith organ, which has two subparts, utricle and saccule, and it has a receptor called macula. So the receptors are crista ampullaris in these three semicircular canals and macula in the otolith organ. Now this maintains the balance of the body in the posture and influence gravity and movement. Next, there is the cochlea. As you can see, that cochlea, it plays the role in hearing, and it has several sensory hair cells in organ of corte. Now the functioning of cochlea is complex, and for understanding the functioning of cochlea we have to take a view of the transverse section of a part of cochlea, and it appears something like this. As you can see, now this is the oval window through which stapes is attached. There's the auditory meatus, tympanic membrane, malleus, incus, stapes. There's the oval window. Now this cochlea, as you can see, this is the bony membrane of the cochlea that we have studied, bony labyrinth, and this is the membranous labyrinth in the cochlea. Now bony labyrinth is covered in perilymph and membranous labyrinth in endolymph. Now the voice travels from stapes to perilymph, and vibrations over here causes vibration in the endolymph, and then it is taken up by the organ of corte. The functioning we will study later. It has three parts actually. This part is called, the upper part in which perilymph is filled, is called scala vestibuli. This is the cochlear duct, and this is the scala tympani, which also has perilymph. As you can see, scala tympani is just continuation of scala vestibuli. To understand how cochlea works, we have to first take a section of this cochlea, and it looks something like this. So as you can see, this is scala vestibuli and this is scala tympani, and both of them have perilymph in them. As you can see through this diagram, both of them have perilymph. This is the bony labyrinth which covers the more delicate membranous labyrinth membrane. This structure is called tectoral membrane, and this is called the organ of corte, which basically helps in hearing. As you can see, this is the organ of corte which goes through the cochlea. This is the cochlear duct. As I've already said, this is the oval window through which the sound goes, and it comes out through round window over here. I mentioned the oval window, this is the round window. Now, the organ of Corti, this is the organ of Corti, and it is located in the basilar membrane and in contact with this part which is called tectoral membrane on the upper part. Now, this contains sensory hairs which are present in rows on the internal side of organ of Corti, and these act as auditory receptors. So sensory hairs in the apical part, they possess stereocilia, and above them there is a thin elastic membrane that you need to remember called tectoral membrane. I have already mentioned. Now just what you have to remember is that tectoral membrane is made up of thin elastic membrane. And as you already know, the basal part is in close contact with afferent nerves which are forming the auditory nerve. So this will be the auditory nerve. So what happens is that because when there is a vibrational disturbance in the perilymph which is present over here in scala vestibuli, in scala tympani, this causes vibration in the endolymph, and the vibration in endolymph causes vibration in the tectonic membrane, and the vibration in tectorial membrane causes the hair cells to detect that vibration, and these hair cells they are connected in basal part with afferent nerve fibers which form auditory nerve, and this auditory nerve takes the signal to the brain. So this is basically how hearing is conducted, and as I've already said that balancing is conducted in three dimension by the three semicircular ducts which are present in right angles to each other, and they have three sensory parts which I've already said, that crista ampullaris, three crista ampullaris in each semicircular canal, and then of course there is the otolith organ which has utricle and saccule, and that has one sensory organ which is called macula. Macula is influenced by gravity and movements, and the crista ampullaris help in balance of the body and posture. This completes this chapter, human ear, and in the next video we will go over the gaseous exchange in humans, which is a short chapter.